Hello, I'm Mr. McKenzie, and welcome back to our last part of our notes on chapters 25 and 26 on the history of life on Earth and phylogeny. We can look at macroevolution, which is a broad patterns of evolution. It's going to take long periods of time, so it's going to take millions of years um, for the evolution to occur. So, an example might be like where terrestrial vertebrates have emerged from a series of speciation events. It could also be something like where we're looking at the horse evolution and we're looking at different species of horse like creatures that have evolved over the last 60 million years. And you can see like the differences between whether it's going to be what it's the feet are like, or what it's going to be like the teeth and the jaw, because of the foods that they're going to be foraging on. Where horses started more in a woodland creature, we can see here with the toes, the four toes, into a open plains, open runner, where they're going to stay on their tiptoes. Conditions for early life that made life possible for life, or early Earth that made life possible. Simple cells developed in four sequences. The abiotic synthesis, meaning like, and we'll talk more about these as we go too, where small organic compounds, molecules have come together. Joining, number two would be joining small molecules into macromolecules. Three would be packaging these molecules up into what are called protocells. And then four, the origins of self-replicating molecules where they can replicate. We've done this a lot by either absolute dating, where we look at radiometric dating, where we use radioactivity, such as carbon-14 or potassium, or one of the radioactive types of elements that we can look at in minerals. Geologists are the people that study these too, and they can study all of these kind of occurrences. So, how did life start? That's our big thing. We look at this abiotic synthesis that we were talking about of macromolecules, and we know the age of the Earth is right around 4.6 billion years old. It's very old. The first water was believed to be found on Earth around 4.4 billion years. Could have even been earlier because of the striking of asteroids um, into the actual Earth. We detect this from minerals such as zircons, and we can see these water molecules in there. Most of the water is believed to come from meteorites. Um, First life forms about 3.8 billion years ago. About 800 million years of no life was occurring at the time. So, the time the Earth started, 4.6 billion to about 3.8 billion, there's about 800 million years. How did life arise? Well, small organic molecules are synthesized. So, first, you have to have those kind of coming together. So, conditions have to be just perfect for this. Small molecules and macromolecules and proteins and nucleic acids are going to start to form up. So, just like everything where we're talking about a chemical reaction and their chemical affinity, the same thing is going on here. And then they have to be packaged into these protocells, and these are what I have over on the right-hand side, these protocells and what they're going to look like, where they're just like small droplets. They have the lipid bilayer. They have some proteins that are going to be in there, and they have a coated capsid protein that's carrying RNA inside. And these RNA were going to work as kind of a ribozyme um, in there too. In 2009, researchers found that these things that can spontaneously occur and be a simple precursor of molecules. And they did this by dripping amino acids or RNA nucleotides in the sand and clay, and they spontaneously form these polymers for each molecule. These polymers could act as catalysts, so because the RNA itself is a simple catalyst, they can act as catalysts with the ribozyme. The synthesis of early compounds, how did life begin? Well, we talked about is how old it is. The first atmosphere was very, very little O2 in it. It had a thick water with large levels of nitrogen, carbon dioxide, methane, ammonia, and hydrogen. And as the Earth cooled, the water vapor started to condense as much of the hydrogen escaped in space. Now, remember, this did not happen overnight. This happened in a long period of time. In the 1920s, there was a hypothesis from A.I. Alpern and JBS Haldane, and they independently came up with the ideas that the atmosphere was adding electrons to the environment, meaning it was actually it was reducing and forming organic compounds. And the energy they thought would come from lightning or UV radiation. So we push forward, and then there's this Miller-Urey experiment in 1953. And the experiment was done to test if there any validity to this hypothesis. And they found that there was a variety of amino acids that were formed. However, there was some evidence that suggests that ancient atmospheres were made up of mostly carbon dioxide and nitrogen, neither reducing or oxidizing. So what they did was they just test this atmosphere and they produced organic molecules. They did that, and these are kind of their testing agent that they had on what it looked like that they were using. Scientists believe today that organic compounds may have been produced near volcanoes where molecules are reduced, and the molecules are reduced. And in 2008, numerous amino acids were found using modern equipment near volcanic eruptions. And then there's something, the Murchison meteorite, which was a 4.5 billion year old meteorite that landed in 1969, and it contained 80 different amino acids, and it was rich in iron and in water too. These are some more protocells where we can see what protocells are like. We also use the fossil record. It was used to reconstruct history. Use sedimentary rock, right, where we have the layers of them, where we're looking at sandstone, limestone, shale, and you can see those to the left here. We're looking at just the different sediments that would happen and what we would have in each one. And then we can have some things that'll mineralize or make a hard body structure, much like any kind of fossils that we're going to find within there. Then there's organic, can be rare ones. This is a piece of amber right here, and that amber is containing in there, you can see an insect that's stuck inside of there. They're rare, but they are found. Also tar pits, where we can look at those. These are the La Brea tar pits in Los Angeles, and you can see where organisms would get stuck in there, whether they're insects or large critters, and then be attacked upon by other predators, and then those things will get stuck, and so on and so forth. We can also see here that there's an incomplete record. Many organisms aren't preserved. Some fossils were destroyed, of course, with whatever glaciation or other volcanic activity, and some are not yet found. Here's the age dating. This would be if we're using to determine fossils, we can use something called index fossils. These index fossils, certain fossils are going to be found in certain types of rock material, and we're looking at sedimentary rock. So that's one way. That's actually known as relative dating. And then we can also look at where we're actually using radioactive decay of isotopes. And this one we're going to get actual dates on here. This one is radiometric dating. So we're using just the radioactive decay, whether it's going to be alpha decay or beta decay. We're going to be looking at that to make new elements. New elements are going to be formed from this in radioactive decay. Half-life is what we're going to use here. The half-life of something is how long it takes for half of the material to decay into a different type of element. So it goes from a radioactive or an unstable element where the nucleus is unstable to a stable form of that, which would be a different element. Something we could see on that would be we could look at using this alpha beta decay. We could look at, if I go back, we can look at carbon 14. Carbon 14 has a half-life around 5,700 years. It decays into nitrogen 14, which is non-radioactive. And this would be beta decay, where we're looking at the nucleus then here would be emitting a electron. So if we're looking at a neutron here would be kind of a proton and an electron that would be in there, negative material. And this negative particle emits out of this, turning it into a proton. So it goes up from being carbon, which is, carbon 12 is normal, having 6 protons in it. It would go to nitrogen having 7 protons then, because the neutron would turn into just a proton on that part. When we're looking at half-life, we're thinking about how long it takes for, if I have something that's carbon 14, and in 5,700 years, half of that is going to be emitted, and I'll be left with carbon 14 on this side. And on this side, I'll be left with nitrogen 14, which is the stable form of nitrogen. And then in another 5,700 years, half of that will be changing into nitrogen 2, and so on and so on and so on. Here is just, when we're looking at different fossils that are coming through, we can look at from the past to the present, where the rock material is going to be laid down, and then different fossil material is coming through on this part. Some key life events, you can see the first organisms. They're roughly going to be about 3.6, 3.8, somewhere in there, 3.3. You see all sorts of stuff billion years ago, where you see the first single-celled organisms, and then you see the first eukaryotes coming in about somewhere about 1 billion years to 1,000,000,003 years on there. And then you can see the first multicellular organisms, and then the colonization of land animals or land plants even. This takes you can see it takes a long period of time for things to evolve and things to go. Once evolution gets started, things get rolling a little bit faster though on that. So you can see O2 was accumulating here. It did accumulate well. And then we're going to find out that plays a role in something here too. And then as we check through it, we can see all of our others. And humans just 200,000 years ago. We've been very recent. We talked about the symbiotic theory, the ambisymbiont theory, but we're looking at how mitochondria and plastids were thought to be prokaryotes living in larger cells, like eukaryotic cells, and would engulf them and then utilize them. The evidence, of course, replication by binary fission for both of those. They have their own DNA in them. The ribosomes are made of proteins, right? They have those too to make proteins. I'm sorry, ribosomes make them. And then enzymes similar to the living prokaryotes are within those structures. And then they have a double membrane system, as we talked about before. We've hit that many times. Pangea is a supercontinent. We use this too to analyze some things. We've talked about Pangea last chapter a little bit in the activity that we had you do, but you can see at different times everything was at a different place as we go in the past. We know it's about 250 million years ago. Donald Drift explains this, but I like to call it plate tectonics. More explains this. The movement of continental plates through plate tectonics, the actual plates are moving and adjusting, and they're still moving today, just as they have been in the past. It's not like we're static all of a sudden. We've had many mass extinctions too, which add to the diversity of life. And then even though the extinctions drop, we see things will have to evolve and change, but it takes a long period of time. Even some of the extinctions took millions of years to occur. So these major periods of the Earth's history end with a mass extinction. Each end of a major period ends with a mass extinction. And new ones begin with adaptive radiations, and we'll talk about this as we go. So there have been 5 mass extinctions. Some take millions of years, some like the end of the Devonian right here, about 20 million years for this all to occur. And organisms die off, leaving exposed niches for the survival of species to evolve and take over. And this will take, like we said, millions of years. 99% of all organisms that have been on the face of the earth have ever been alive are now extinct, and we're a member of that surviving 1%. Okay, so we have at the end of Ordovician. Now, I don't expect you to memorize these, and no matter wherever you look, these might vary slightly. Um, but the end of the Ordovician, the end of the Devonian, the end of the Permian, the end of the Triassic, and the end of the Cretaceous. Okay, you can see this one was the largest one at about 251 million years ago. 96% of the species died from that one. Okay, so Precambrian time is the 90% of the Earth's history. It's the earliest. It's going to be this whole part back here from when it first starts, and it's going to be the first organisms are going to start forming during this time at that 3.3 billion years ago. Archaeobacteria and cyanobacteria are coming into play at about 2.2 billion years. First pollution crisis, where we actually have oxygen forming at high rates. We did have oxygen, but now we get high rates of this into the atmosphere. We go from 1% to about 15% of the atmosphere, and that was toxic to most bacteria, and most of them died off. But some used it to O2 to produce energy, and those are what we feel became the mitochondria, right? Um, First plants evolved somewhere around 900 million years ago from photosynthetic prokaryotes such as cyanobacteria, and our first plants were relatively, simple. They were non-vascular plants. There were some vascular plants that came later, but you can see that they were just like real small, almost like some mosses or in that portion of it. The first animals we're looking at about 650 to 500 million years ago. Again, you don't need to know dates. Just need to know that things move into various forms, and our evolution takes a long period of time. Um, it gave way to the evolution of skeletal organisms. These weren't skeletal organisms; they were very, very almost plant-like, and they lack skeletons. The fossils found on every continent except Antarctica, and they died due to changes in the sea level. So at the end of the Ordovician time, this was the first mass extinction that we have for you at about 440 million years, 443. It's the third largest mass extinction. Most sea creatures like graphlites and trilobites, and you can see those up in here, graphlites, trilobites, and then you got some brachiopods here that look like what we'd think of as like a shell that we would find from clams, and they weren't. They died because of increasing the increased ice and lowering the sea level. So our temperatures changed. Um, Silurian period comes after, and this leads into many firsts. All of a sudden, now we see fish starting to appear, like jawless fish, like this lamprey here is a Agnatha jawless fish, and this hagfish, which both of them are around today, are in that same category. Also, ancestors of spiders and insects and vascular plants and fungi are all playing a role in this portion. It began the Devonian period. We at the end of the Devonian, we see another mass extinction. So this would be the second mass extinction. About 75% of the life died out. The reason for this was again plant plants that produced a lot of O2 in the atmosphere, released nutrients into the ocean, and these nutrients then caused almost like an algae bloom. It caused living things to really explode, consuming water or consuming the oxygen in the water. So then everything was going to start dying out. Remember, organisms were living in the water. There were no terrestrial organisms here at this part. It ended about three. There were, I shouldn't say that. There were some terrestrial organisms. I'm sorry. But at the end, at about 359 million years ago, then terrestrial environments weren't as affected as much as the aquatic or the marine environments. So the smaller organisms that could survive on land, much like a lot of like amphibian-like these tetrapods survived, and they were moving from the sea to the land, and this kind of forced them in this evolution. This 20,000 or 20 million years of evolution for them to evolve and move through. The largest mass extinction is the end of the Permian, and the Permian we're looking at this at about 251 to million years ago. About 96% of the species died. So 96% of those that were living died off. Today, all organisms are descendants of the surviving 4%. So it affected corals, took 100 million years for new corals to be forming. Rocks from this time period show no coral reefs or coal deposits. The cause of this was large volcanic activity emitting toxic gases and fumes, causing the oceans to become highly acidic. It also depleted the ozone layer. So all of a sudden now you have deadly UV rays that are coming down through the atmosphere now, coming from the sun, and it's basically burning up the Earth's surface. And it caused a lot of wildfires. In fact, we see this like charcoal gap where we don't see carbon. We have lack of carbon that we're finding. That's why we don't find the coal deposits, because everything just kind of burnt up at this part. The survivors here were going to be a lot of fungi, probably living on the dead material. Also, smaller dinosaurs, along with snapsids. These are relative to mammals, and these are they have a little hole right behind their eye socket. These holes, that's what really makes them, and they basically evolve then into mammals. They look a lot like, as you could look at this, they look a lot like a dinosaur or a reptile, but they're more closely related to a mammal on this part because of this hole behind the eye socket. Okay, and there are just some what they were like. The larger ones start to die off, and some are predatory, but they start to die off. Sorry. The end of the Triassic period, about 201 million years ago, during the Mesozoic age. So this is all the ages of the reptiles, the Triassic, Jurassic, and Cretaceous periods. But we see about 80 at the end of the Triassic period. We see about 80% of the species die off. We see a mass extinction here caused by basaltic lava flows. Again, a lot of volcanic activity. Killed off the conodonts, which are these small fish-like creatures, which we'd see right here. Here's some of the teeth of them, and here's a fossil of them too. Um, but and small reptiles. Small reptiles were also eliminated here. But now it opened up a niche where now dinosaurs could really thrive, and so they could start to rule. You go lead into the Jurassic period, which is going to be the dinosaurs, and then, of course, at the Cretaceous, the small mammals start to forming here too. Sorry about that. Went a little bit too fast. The end of the Cretaceous is where we see the dinosaurs go extinct. The mass extinctions at the end of the Mesozoic era, the KT extinction, they call this one, or Cretaceous tertiary extinction. Over 50% of all life died, including most dinosaurs. Survivals include mammals, and the surviving branch of the dinosaurs, the birds, are going to be in here. We're going to see birds that will form. And what caused this event? Well, we believe that it's the asteroid, that hit the Yucatan Peninsula, as well as volcanism. All of these cause the ocean levels to decrease due to extreme cooling. We also know that it caused, because of all plumage of atmospheric dust, it would now block out the sun and would cause this extreme cooling too. And the Cenozoic era is what we're in today, 65 million years ago to the present, the age of the mammals, and we can see where mammals are, they just started to proliferate. The pace of evolution, there's something called gradualism. It's just accumulation of small changes. And then there's punctuated equilibrium, long periods of stasis, meaning nothing's happening, followed by rapid change. Now, rapid in the relative is a relative term. Is that we're looking at thousands, we're looking at millions of years. We're looking at time, but rapid in Earth's history. It stabilizes and it oscillates the selection responsible for the stasis. Punctuated equilibrium came up with by a guy, by two people, gentlemen, Niles Edridge and then Stephen Jay Gould, and these bursts that would occur for very short periods of time and caused by an extinction and an open niche. The problem is that you're probably going to see both gradualism or natural selection kind of, where it's going very, very slowly, and punctuated equilibrium are working together. There's something called Evo-Devo too. Again, this was something that was brought up by Stephen Jay Gould and his colleagues. Um, Evolution is new forms from changes in DNA, and the regulation of these genes causes differences in development. So it's a study where how these organisms are going to develop important evolutionary biologic for several reasons, and that's to survive. And we can look at this whether we're looking at you know um, the forelimbs like we've talked about before, the homologous types of structures, and we can look at how they've formed, whether it's going to be the bird wings and wings of a reptile and wings of a bat, a mammal, the dolphins flipper and the seal flipper, a dog's forelimb and the sheep forelimb or the human forelimb, and how everything kind of evolves through this, having certain similarities. We can also look at something where we're looking at these structures here on these two, maybe when we're looking at a reptile versus a salamander, and the reptiles can push themselves up. They also have to have larger pectoralis muscles so they can push themselves up, but the bones have to be slightly different. So you're going to see this evolutionary change that's going to happen. So the bones aren't keeping it so low to the ground. It's able to push itself up. Plus, it can run faster within that. And then there's going to be these genetic markers that are going to come into play. We're going to see that too. And that's just Evo-Devo, or evolution and developmental biology together. We'll be looking at those things. Um, uh, heterochrony, evolutionary changes in the rate of developmental events. It's a genetically controlled difference in the timing of developmental events. So for what we're looking at, this is where the genes are controlling how organisms are going to develop. So it might slow down the development of like the body. It might alter maturation or reproductive systems. We need to look at the life history, the ontogeny of an organism. So let's say, for instance, you're looking at like an organism such as it could be humans or it could be a chimp. A lot of our skulls look relatively similar, but we look different from how we looked when we were born, and babies look much different than the adult version. And even when you become like maybe you're 18 years old, you look much different than what you're going to look when you are um, 60 years old. You'll have differences in your nose structure or your eyebrows or whatever. Your facial features will look different. You'll start to look. The same is true like a puppy, how a puppy looks all cute and kind of full, and then turns into be a larger dog, and its head shape changes, and all sorts of things change. Um, we can also look at something called pediomorphosis. In this pediomorphosis, the adult retains a juvenile structure. So the adult looks a lot like what the juvenile uh, of that species look like. So that would be where something stays looking like the younger version. It doesn't change. So a puppy and an adult dog would look the same. Well, these are gills on this this structure here, which would you would find the gills on the same thing. That's like mud puppy on type of a juvenile of the species. So homeotic genes are master regulators. They determine the location and the organization of body parts. Now remember, this is all kind of the part of this Evo-Devo, right? These genes regulate the development of anatomical structures in organisms. So they actually regulate where these are genes that are going to say, hey, let's develop forelimbs, or let's develop hands, or let's develop feet, whatever. They're going to be regulating those, where to place those, or let's develop, if you're a bird, wings, or if you're a bird, feathers. The Hox genes are responsible for directing the body plan, meaning like from the head to tail um, axis of an animal, where these are going to be placed in the certain order. So making sure the structures are in the correct part of the body, so the correct places that would occur. So these would be the Hox genes, placing them in. We would see a Hox gene expression of whether it's going to be a human placing in these four limbs that are going to be placed in here along that head to tail axis. It could be where gills are going to be placed on fish, or where numerous items. You can just go on and on for this. But it's really a lot that we would look at within that portion on here. We can also see where Hox genes, if they have a mutation in there, how it can change into a different species. So now we see a mutation in a Hox gene. It's something like on this, this fruit fly here, Drosophila. You could see that its legs, where it places its legs to something like an Armadillo, you can see where the legs are going to be in the middle. So uh, each one of those would be from a mutation in a Hox gene. Uh, Exaptations are where something evolves and it's co-opt to a different function. It's not following its original function that it might be used for. But like bird feathers, they really formed for thermal regulation to keep it warm. It wasn't used for flight. It didn't build the flight muscles or anything like that at that time. But then over time, as it started to evolve, flight came with it, and they were great for flight. Um, the same thing you could think of sutures in the skulls of humans. They're there for the growth as your brain grows, and the sutures then kind of come together to protect your brain. But for our brains, which are big, and our heads, which are big upon birth, it also, is able to keep the head where it can be kind of pushed and takes different shapes for birthing purposes. So that we could look at too. There's two parts. That's in verse 25. Let's look at chapter 26, which is pretty easy because we looked at a lot of these. In chapter 26, what we need to know is the taxonomy. So taxonomic categories and how they, they relate. Systematics that's used to develop phylogenetic trees. And then three domains of life, including their similarities and their differences. So systematics is just classifying organisms and determine their evolutionary relationships. Systematics is going to include taxonomy, which is just classifying things, and then phylogenetics, which is the evolutionary history. Here we'd be looking at something like evolutionary relationships. We've seen this in the past. We're just looking at just a little evolutionary trees on here. And fossils and morphology, homologous structures, right, that we talked about. Molecular evidence, DNA and amino acid evidence, we talked about those. And then who's more closely related? Well, you would say, well, according to these, the fungus would be more closely related to the human because it branched off relatively recent compared to where the branching was for the flowers, right? And you'd be correct on that. So molecular evidence would probably give us less amino acids, changes in amino acid sequence too, from humans to a fungus compared to humans to a flower. So that's within that slide. Taxonomy is just classifying or naming organisms, how we name them. And we start with our domain, and the domain can be three types that we'll see: eukarya, uh, and then the prokarya, and then the archaea that we could find within there. Um, then we get to our kingdoms, our animal kingdom. You have the animals and the plants, and you have the fungi, and you can go on from there, right? And then we have our phylum, and then our class, then our order, our family, and our genus and our species. So each one gets more and more narrow. So you can see that this is bringing it down to more and more narrow until you get that one species that's going to be there. There might be multiple things under the genus and multiple things under the family within. this is showing something. It started by Carl Linnaeus, and really we're looking at what we were talking about and narrowing it down. So it's starting from the domains, which the domain would be eukarya, and then you go to the kingdoms. You can see another domain, archaea, and another domain, which would be bacteria or the prokaryotes, right? And then you can see that we would start going, each one gets less and less and less in number, and it gets smaller and smaller. So this basically is making this triangle like that till we get to the proper species. Whenever we get down to the scientific name, which is including the genus and the species, Panthera pardus, you can see that the P for the genus is going to be capitalized, but the P for the species is always lowercase, and that's always what we do. And even if you look over on the left side, Ursus arctos, you can see like that is going to be a capitalized Ursus, and then arctos is lowercase. These are the three that we would see, eukarya, archaea, and then bacteria. And how are they overlap? Those are the three domains. You should know those. Phylogenetic trees, we've talked about these. They're branching the organisms within evolutionary history. We know that each time there's going to be a common ancestor would be located here at each part where it branches off. And that's basically what we would have within these as we start to branch off for each portion of these. A cladogram is just a diagram that depicts the patterns of shared characteristics in the taxa. A clade is just a group of species that includes the ancestral species and all of its descendants. Shared derived characteristics are used to construct cladograms. It would be something like this. We've seen these again. It's just an evolutionary tree, and we're looking at the cladogram, and we're just looking at what characteristics do they share. Everything above this point right here has vertebral column. Everything above this point right here has hinged jaws. Everything above this point has four walking legs. So you can see if it branches earlier than that, like the tuna, doesn't have four walking legs, but everything after that has four walking legs. Same with the hinged jaw. We know that the lamprey doesn't have a hinged jaw because it's part of the agnathus or the jawless fish, but everything after that has a hinged jaw. And that's what we're looking at within cladograms. This is monophyletic and then paraphyletic and then polyphyletic groups, meaning how are they going to share? What are they going to share? Are they going to share one, two, multiple things that they're going to share within them? Constructing a phylogenetic tree, you just indicate, well, like what we've talked about before. What are they actually sharing? And you can see by looking at, are they sharing one? So one indicates a character is present. So each one showing that they have that character present, that's going to um, make them closer related. So a vertebral column. So each one of those has a vertebral column after, whether it's a lamprey or whatever. Hinged jaw, well, each one of those has a hinged jaw after. So as we're going up that scale, we can see that. Okay. Then various layouts of them. They could be circular, they could be a rooted tree, they could be an unrooted tree. There's various layouts that you can have with planograms and clades. There's something called the principle of maximum parsimony, which is just the simplest idea or the simplest, have the ones with the fewer changes in DNA or the fewer changes in their genetics for the tree, keep that. That's probably the right way. It's the simplest way. So just keep them simple. There's also molecular clocks. Some regions of the DNA appear to evolve at a constant rate. So as we look at this, you can see on the bottom, we have this constant rate that we're looking at here, as in these regions with molecular clocks. And it estimates the dates of past evolutionary events. So remember, it's an estimate, it's not an actual date. Like the origin of HIV infections in humans in the 1930s, and you can see how that is going and how that changes. And we'll see that with any type of disease that we're looking at. Oops. Um, and the last slide is just about systematics. We're focusing on biological diversity within it. We focus on phylogeny, which is just, the history of these things and what they're related to. Then taxonomy, we can see that biological diversity and taxonomy, identification of species, um, looking at the taxonomy with the domain, kingdom, phylum, class, order, family, genus, species. And then we can look at binomial identification. We're looking at binomial, meaning it's two names, genus and species. Then we can see that phylogeny is just classifying, again, classification of material, and how that goes together with taxonomy. They can go back and forth. Phylogeny, we might look at cladistics or making those clades. Homologous similarities in classification too are those homologous structures. Fossils, what the fossils look like and how they indicate them. Molecular homologies might have molecular structures or molecular DNA that's similar. And then morphology, the shape or what they look like or how they're using those. And that is it. And I hope everybody, if you have any questions, just feel free to get a hold of me. Thank you.